Hello everyone, welcome to another IVIMS webinar. I'm Kübra from IVIM Technology and I'm glad you could join us today. In this session, we will be discussing visualizing next generation nanomedicine from whole body imaging to intravital microscopy. We will explore the differences between macro or organ level imaging techniques such as bioluminescence, PET, CT, and MRI, and micro or cellular level imaging, specifically intravital microscopy in our case. As you listen to Jenny's presentation, feel free to visit our website ivimtech.com and our shop page ivimtech.shop to learn more about the imaging tools we will be discussing today for in vivo cellular imaging in live animals. Now I would like to introduce today's presenter. Due to an unexpected health issue, Michael won't be able to join us today. Instead, Jenny from our technical application team will be presenting. She is the lead operator of our animal experiments for CRO services at IBM Technology in the sales division, following her work with the R&D team. If you have any questions related to especially the surgical procedures or animal preparation, she is a perfect person to provide the detailed insights. So please feel free to submit your questions in the Q&A box below at any time during the presentation, and we will do our best to address as many as we can at the end of the session. Jenny, if you are ready, please go ahead with your presentation.
Thank you, Kubra, for the introduction, and hello, good morning, everybody. I'm Jenny Ohm, Technical Application Specialist here at IVIM Technology. Thank you all for joining today's webinar on visualizing next-gen nanomedicine from whole body imaging to intravital microscopy. And again, we apologize for the absence of our original speaker, Michael Yoon, but I still hope you're all still excited for today's webinar, just as much as I am. So in this session, I will introduce how IVIM Technology's intravital microscopy can be applied in the development of various nanoscale drugs and medicine with various application cases as well. And I will also briefly introduce IVIM Technology's CRO research service workflow. Then at the end of the webinar, we will address any questions you may have during our Q&A session. And without any further ado, let's begin with today's webinar. Okay, so as many of you may know, the development of a new drug typically takes between 10 to 15 years and requires an investment cost of approximately 1 to 2 billion USD. And despite this enormous commitment of time and resources, unfortunately, around 80 to 90% of drug candidates fail during the clinical stages. In fact, only about 5% of drug development projects ultimately succeed in reaching the market. The drug development process is generally divided into 4 main steps. First, target identification and lead discovery. Second, preclinical testing. Third, clinical trials. And finally, regulatory approval and post-marketing surveillance. Among all the new drugs that are put into development, around 60% of the drugs that fail typically fail when transitioning from the preclinical phase to the clinical phase. There are many reasons for these failures, but a major factor would lie in the discrepancy between the preclinical and clinical trial results. This is largely due to the limitations of conventional preclinical approaches, such as in vitro assays and endpoint-based methods, which often fail to accurately reflect the drug's behavior and efficiency in living organisms. So as a result, the drugs may not reach the intended tissues effectively or may not act precisely on the desired targets or even cause unexpected side effects, all leading to failure in the clinical trials. Ultimately, to address these challenges, we need new technologies that can allow us to precisely observe and understand how drugs behave in real living biological systems and organisms. And one powerful technology that can help overcome these limitations is intravital microscopy. So what exactly is intravital microscopy? Intravital microscopy, or IVM, is a high-resolution imaging technique that allows for real-time observation of cellular and tissue-level dynamics within living organisms. And traditional imaging methods, such as in vitro or ex vivo imaging, approaches typically rely on fixed tissue slides or cultured cells. As a result, they lack the ability to capture real-time biological and physiological changes and do not fully reflect the complexity of the in vivo environment. While modalities such as PT or MRI also offer amazing in vivo imaging capabilities, they primarily provide insights at the organ level and lack the spatial resolution required to observe individual cells in action. And in contrast, intravital microscopy enables the real-time tracking of drug behavior, biological responses, disease progression, and etc. at the single cell level in living animals. So it's a very powerful tool for visualizing various biological phenomena, including drug delivery and distribution, cell-to-cell interactions, immune responses, and so on, all with high spatial and temporal resolution. And as mentioned before earlier, one of the most traditional methods used to analyze drug responses in the drug development process is the endpoint method. This approach involves sacrificing animals at specific time points to collect tissue samples for histological analysis. However, this method has significant limitations. It cannot adequately capture the variability of drug responses between individual subjects and falls short in providing a precise understanding of drug distribution or mechanisms of action within the organism or animal. So as a result, it becomes quite difficult to accurately evaluate a drug's efficiency, increasing the likelihood of failure in clinical trials. In contrast, intravital microscopy allows for real-time longitudinal imaging and tracking of drug responses within the same individual subject for over extended periods of time. This makes it possible to precisely determine how effectively the drug reaches the target tissue, as well as the drug's pathway of action, and determine whether or not the drug causes off-target toxicity in any non-target tissues. Furthermore, because repeated measurements can be taken from the same subject, intravital microscopy can significantly reduce the number of animals required for experimentation, which leads to savings in both time and cost. So to acquire more precise, reliable, and replicable data during in vivo imaging with intravital microscopy, IVM technology has established an optimized fluorescent labeling protocol specifically designed for in vivo applications. This involves conjugating fluorescent dyes to antibodies and administering them via intravenous injection. Most importantly, the injection dose is precisely controlled at between 25 to 33 micrograms, which minimizes variability caused by dosing differences and ensures both accuracy and reproducibility of the experiment. Moreover, imaging can begin immediately after injection, significantly enhancing experimental efficiency and reducing time overall. So this optimized labeling protocol plays a very key role in ensuring high quality data acquisition and maximizing workflow productivity. So now let's take a brief look at some modalities used for in vivo imaging, as well as taking a closer look at the distinct advantages of IVM Technology's intravital microscopy system. So when looking more closely at in vivo imaging, commonly used tools such as the X-ray, PT, CT, MRI, and even IVIS have played a very important role in providing whole body or organ level information in animal studies. These technologies are highly effective for visualizing overall anatomical structures, physiological functions, and the general distribution of cells or drugs within the body at specific time points. And here, intravital microscopy, on the other hand, complements these modalities by enabling cell-level imaging in living animals, as well as real-time observation and analysis of dynamic phenomena within live tissues. This capability makes it possible to visualize the targeting and effects of new biotherapeutics and treatment technologies directly inside living organisms, adding a layer of detail and functional insight to traditional in vivo imaging approaches. So to advance research in fields such as immunology and drug delivery, it's essential to understand how and why biological processes occur within the living body at the cellular level. So IVIM Technologies' Intravital Microscope enables dynamic, real-time visualization of cellular responses and disease progression in vivo. This capability provides critical insights that help guide the development of effective therapeutic strategies. Furthermore, by using live animal models, researchers can track the pharmacokinetics and pharmacodynamics of drugs over time or in real time, which is a powerful advantage in preclinical development. These functions also support the generation of highly reliable data that meet the regulatory standards required for drug development. And as shown in the examples here in the slide, on the left, we see pulmonary vascular blockage in the lungs induced by inflammation in a sepsis model. And on the right, we can see that the distribution of exosomes and anti-cancer agents, as well as the dynamics and migration of transplanted stem cells, have been imaged and tracked. In addition to its imaging capabilities, IVIM Technologies' Intravital Microscope is provided as an all-in-one system for very easy usage. This all-in-one design system simplifies installation and maintenance, and it eliminates the need for a darkroom, allowing users to focus entirely on imaging and performing experiments without any constraints from space or the surrounding environment. The system incorporates several advanced features that ensure exceptional in vivo imaging performance, including a high-speed polygon scanner, a real-time temperature monitoring system, and a motion compensation technology. We also provide separately an AI image denoiser program or software, which is an AI-powered noise removal software. So the motion compensation feature corrects for organ movement, enabling sharp and accurate imaging even in organs with very significant motion, such as the heart and the lung. And our real-time temperature monitoring system here allows researchers to track and adjust body temperature during long imaging sessions, ensuring compliance with animal welfare standards and minimizing physiological variation for more reliable data. Lastly, here our AI-based noise removal software enables true quantitative analysis by effectively removing noise while preserving the original fluorescent signal emitted from the organ or sample, ensuring very clean and precise data sets for further data analysis. Furthermore, IVIM Technologies' Intravital Microscope is designed not only for short-term, but also for long-term imaging, ensuring stability throughout extended experiments. To support this, we provide a wide range of customizable imaging windows and chambers that facilitate stable, long-duration imaging across various organs. These chambers also enable real-time pharmacokinetics and pharmacodynamic analysis by allowing direct visualization of drug movement and distribution within the body over time. The current slide here shows examples of some of the most widely used window and chamber designs of IVIM technology. If your research focuses on a specific organ or requires specialized conditions, we are more than happy to assist with the custom development and optimization of imaging windows and chambers tailored to your experimental needs. If you are interested or have any inquiries regarding this, please feel free to reach out to us to discuss further on how we can support your research goals. So by using IVM Technology's intravital microscope, we can visualize various organs in real time in living animals. This enables direct observation of a wide range of biological processes, such as immune responses occurring in vivo, tumor progression and metastasis, drug delivery, mechanism of action, and much more. Let's take a look at a few representative examples that I've put in the slide here. So firstly, depending on the drug formulation, we can observe differences in immune activation induced by various drugs. We can also evaluate therapeutic efficiency during cancer metastasis, or we can visualize fibrotic targets for NASH, or also known as non-alcoholic steatohepatitis therapeutics, and assess the delivery and efficiency of various drug formulations. These examples illustrate how intravital imaging offers powerful and mechanistic insights across many therapeutic areas, from oncology to immunology and even fibrosis. Now I'd like to walk you through a few experimental cases that were conducted by using IVM Technology's Intravital microscope. These examples will give you a deeper look into how the system is applied in actual research settings and how it can provide critical insights across different stages of drug development. So the core mechanism of exosome-based therapeutics lies in their natural ability to be able to transport bioactive molecules, such as proteins, nucleic acids, and small molecules, directly to specific target cells or tissues. Leveraging their endogenous origin, exosomes can cross physiological barriers, evade immune clearance, and deliver therapeutic cargo with very high precision. Therefore, directly observing how efficiently exosomes reach and release their payload at target sites is critical for understanding their delivery efficiency and mechanism of action. In the following experiments, we visualized the real-time translocation, delivery, and distribution of therapeutic exosomes in the abdominal organs. Furthermore, we investigated their biodistribution patterns within the mouse brain to assess targeting specificity and delivery outcomes. So for the first experiment, we used a lyse-M GFP transgenic mouse to visualize the immune cells. And we also used IVM Technology's labeling kits that utilizes the Ly6G antibody and CD31 antibody to further visualize neutrophils and the blood vessels, respectively. Then the mice were divided into two separate groups, and one group was injected with PBS as a control. Then the other group was injected with LPS, or also known as lipopolysaccharide, to create the treatment group model, or the sepsis model. After this, the drug-filled exosomes were labeled with a red fluorescent dye, which were then injected into the mouse through intraperitoneal injection. Once the modeling step was complete, we conducted intravital imaging at three main time points, which was before exosome injection, 10 minutes post-injection, and finally 30 minutes post-injection. And the objective of this experiment was to visualize the effective delivery of the drug-filled exosomes and its biodistribution in the liver and spleen using intravital microscopy. So here in the slide, we can see the results of the experiment in more detail. First, on the left is an image showing the mouse securely mounted and prepared for in vivo imaging of both the liver and the spleen, ensuring stable positioning for clear visualization. On the right are the results obtained from the sepsis-induced mouse group. At the 10-minute post-injection time point, we could begin to detect the integration of exosomes into immune cells, specifically neutrophils and macrophages. This early uptake pattern became more distinct and pronounced at the 30-minute time point, where the fluorescent signals clearly outlined the interaction between the exosomes and these key immune cells. Together, these findings highlight the rapid targeting and cellular engagement of the exosomes within inflamed tissue. If we take a much closer look at the results of each time point, you can see here at the 10-minute time point, the exosomes, which are labeled with red signal, have begun to become internalized into the neutrophils and the macrophages, which are labeled here as green and blue fluorescent signals. And at the 30-minute post-injection time point, the exosomes were almost completely internalized into the immune cells. This pattern was maintained throughout various spots, even after the 30 minutes post-injection time point. As you can see here on the right, we also conducted in vivo imaging at 1.5 hours post-injection and could observe similar localization of the exosomes into the immune cells, showing successful delivery of these drug-filled exosomes to their target immune cells. Next, we also wanted to see if the same localization occurred in the spleen or if there was a different distribution pattern. So on the left is the control group, which was injected with PBS, and on the right is the sepsis group induced with LPS. As a result, we could see a significant difference in localization and distribution between the two groups. In the control group, we could see that the exosomes were dispersed or quite spread out in the spleen and weren't exactly internalized in clusters inside the target immune cells. Whereas on the right, in the LPS-induced group, we could see that the exosomes were distributed in clusters very near or almost inside the immune cells. And if we look at the exosomes in the LPS-induced group, we could again see that there was a notable localization of the exosomes within immune cells, along with numerous exosome clusters surrounding them. So this spatial pattern clearly demonstrates the interaction of these exosomes with their intended immune cell targets, thereby confirming their successful delivery. So in summary, intravital microscopy enabled real-time cellular-level visualization of the targeted delivery of the drug-loaded exosomes in a live mouse model, which then confirmed the effectiveness of the delivery system. And as mentioned before, directly observing how efficiently exosomes reach and release their payload at target sites is critical for understanding their delivery efficiency and mechanism of action, especially across challenging barriers like the blood-brain barrier, or also known as the BBB in the brain. In the following experiments, we aim to visualize such delivery and biodistribution in the mouse cortex using intravital microscopy. So in this slide on the left is a mouse surgically prepared and mounted for in vivo brain imaging. You can see the cranial imaging window implanted here on the mouse. And for this experiment, we used BLBC nude mice, and after surgically implanting the cranial imaging window and going through a 2-week recovery period, fluorescently labeled exosomes were intravenously injected. And also, again, using IVM Technologies labeling kits, we injected fluorescently labeled antibodies to visualize the blood vessel structure of the brain. And intravital microscopy was then used to observe real-time exosome distribution in various spots in the brain. So here we utilized IVS IVs to track the accumulation of exosomes in mice over an extended period. From day 1 to day 22, we observed a progressive buildup of exosomes throughout the mice. These findings are meaningful as they provide spatial information at the organ level, confirming successful accumulation of the exosomes. However, when these results are combined with intravital microscopy, which offers real-time, high-resolution imaging at the cellular level, we gain much deeper insights into the dynamic behavior of these exosomes. And here are the results of the experiment. In vivo imaging utilizing intravital microscopy was done at 24 hours post-injection of the exosomes. And as reference, the blood vessels were labeled with a green fluorescent signal, and the exosomes were labeled with a blue fluorescent signal. In areas 1 and area 3 in the mouse brain, we could observe the localization of the exosomes around the blood vessels of the cortex, and the distribution pattern was close to a smearing type pattern. Also, we could observe that not only were the exosomes found surrounding the outside of the blood vessels, but we found out that they were also inside the blood vessels. This suggests that the injected exosomes were diffusing out of the blood vessel into various parts of the brain parenchyma. So these observations highlight how intravital microscopy enables real-time, high-resolution visualization of exosome dynamics and vascular interactions in the living brain. So combined with our earlier IVs data, these results provide spatial and temporal resolution sufficient to directly observe blood-brain barrier penetration and cellular level localization of exosomes in deep brain areas. And in the following experiment, which is somewhat similar to the previous one I just showed you, it also aims to capture the dynamic distribution of exosomes in the cortex. This time, however, we wanted to see if the injected exosomes had the potential to break through the blood-brain barrier and disperse into deeper areas of the brain. So after cranial imaging window implantation, followed by a 2-week recovery period, the mice were injected with exosomes and fluorescently labeled with anti-CD31 antibodies and anti-CD11b antibodies to visualize the blood vessels and dendritic cells in the brain. Imaging was then performed repeatedly at different time points, which were before exosome injection, 2 hours post-injection, 6 hours post-injection, and finally 24 hours post-injection. The objective of this experiment was to observe not only the distribution of the exosomes in the cortex by depth, but to also see whether or not these exosomes co-localize with the dendritic cells found in the brain. Here are the results from the experiment. In these images, the blood vessels are labeled in red and the exosomes are labeled in green fluorescent signals. And at the 2-hour post-injection time point, we began to detect exosomes flowing through the cortical blood vessels, which became much more prominent by the 6-hour time point. By 24 hours, we observed clear diffusion and penetration of exosomes from the cortical blood vessels into the surrounding tissue. And in the areas marked with white arrowheads, you can see that the exosomes have moved from inside the blood vessels to the outside, distributing into the adjacent brain parenchyma. This progression over time provides direct visual evidence of exosome transport and extravasation in the cortex. Moreover, at the 24-hour post-time point, we conducted further analysis to determine how deeply the exosomes had penetrated into the brain tissue and how they interacted with the surrounding immune cells and here in this case, the dendritic cells. So to achieve this, like I mentioned before, we use the CD11B antibody, which is a cellular marker of the dendritic cell to visualize those cells. Then the distribution of the exosomes and dendritic cells were analyzed. So in order to acquire imaging data on the depths of the brain, Z-stack images were acquired at 10 micrometer intervals, starting from the cortical surface and extending to depths of 30, 40, 50, and 60 micrometers. Through this, we were able to assess both the penetration depth of the exosomes and the positioning of the dendritic cells. The results showed that fluorescent signals from the labeled exosomes were observed up to approximately 50 micrometers deep outside of the blood vessels, and co-localization of the exosomes with the dendritic cells were also very evident. So these findings suggest that the exosomes were not only penetrating the brain tissue very effectively, but also had the potential to interact directly with immune cells. This provides very meaningful insight into the feasibility of exosome-based immunomodulation and targeted delivery strategies. So in summary, this experiment goes beyond simple distribution mapping by highlighting the potential for cell-level targeting and immune system interactions, which underscores a much broader significance of these findings. The next and final experiment is similar to the previous one, but here we aim to observe the co-localization of exosome with cancer cells. So in order to evaluate their ability to specifically target those tumors. For the experimental setup, we used BLBC nude mice and surgically implanted them with the cranial imaging window, and again allowed a 2-week recovery period. And after recovery, we injected medulloblastoma cells to establish a mouse brain cancer model. And following this, we administered both anti-CD31 antibodies conjugated with a fluorophore and the exosomes intravenously. In vivo imaging was then performed at 24 hours post-injection, and the primary goal of this experiment was to, again, visualize the co-localization of exosomes with cancer cells in the cortex using intravital microscopy. So here are the results taken at 24 hours post-injection. For reference, the blood vessels were labeled with a red fluorescent signal, the cancer cells with a green fluorescent signal, and the exosomes with a blue signal. In various regions, we could observe that many exosomes were clustered within the cortical blood vessels. Moreover, in areas where cancer cells formed clusters, exosomes were also very accumulated and distributed around those regions. This indicates that the exosomes co-localized with those cancer cells in the cortex, suggesting that they effectively distributed to and bound to their target cells, confirming their targeting efficiency. These findings also highlight the potential of intravital microscopy as a very powerful tool for evaluating nanocarrier-based drug delivery systems and advancing brain-targeted therapeutic strategies. Overall, the experiments that I've presented so far in my presentation demonstrate that intravital microscopy allows us to effectively observe the distribution, movement, and dynamic behavior of exosomes in vivo, underscoring its utility as a highly valuable method for studying targeted delivery and cellular interactions. So in addition to the liver, spleen, and brain imaging results I have shown you, high-resolution intravital imaging can also be applied to a wide range of organs in living mice, including the lungs, heart, pancreas, and many more. This versatility allows researchers to investigate biological processes in diverse systems, all in real time and at the cellular level. Furthermore, iBeam technology not only offers an all-in-one system optimized for high-quality intravital imaging, but also provides comprehensive CRO services tailored to the specific needs of each research project. By combining advanced imaging capabilities with professional research support, we enable scientists to conduct their studies more efficiently and generate more robust data and ultimately accelerate the path from discovery to very meaningful results. So what you're seeing here now are actual customer cases where we conducted CRO services together with IVIM technology. We collaborate with various companies, research institutes, and universities to provide CRO services for both short-term and long-term imaging projects. I'll now briefly introduce the overall workflow of how our CRO service is carried out. So the CRO service provided by IVIM Technology is designed to support researchers from the very first idea to the delivery of the final results, and it follows a structured step-by-step process. We first begin with a thorough consultation, during which we discuss the experimental objectives, design, and specific requirements in detail. This stage ensures that we fully understand the research goals and can tailor the approach more accordingly to your needs. Next, we carry out a preliminary feasibility test. This test is essential for confirming whether the proposed experimental design can be successfully implemented and whether it will produce reliable, high-quality data. By validating the methodology at this stage, we help prevent costly delays or failed experiments later on. Once the feasibility test is confirmed, we move on to the main experimental phase. Here we conduct the in vivo imaging, leveraging our high-resolution intravital microscopy systems and collect the raw imaging data. Following this, we perform careful post-processing and in-depth analysis of the data to ensure that the results are both accurate and very meaningful. Finally, all the findings are compiled into a comprehensive final report, which is then delivered to our clients. For more information about our CRO service, you can reach out to us via the email address shown here, or you can visit our website at www.ivimtech.com for further details. So thank you everybody for joining us for today's webinar. Over the course of this session, I explained why intravital microscopy is vital to the overall drug development process and how it can be applied to drug development. I also highlighted the advantages of IVIM Technology's unique intravital microscopy system and shared some application cases acquired with our system. We sincerely appreciate your time and attention, and that concludes my presentation. Thank you.
Thank you, Jenny, for the nice presentation. Our first question comes from Dr. Yang, who submitted during the registration. But before that, if you have any questions, please feel free to ask them at the Q&A box below. The question from Dr. Yang is, how long does a fluorescence signal last when you label the blood vessels?
So in the case of the blood vessels, they typically last up to 24 hours. So if you would like to image longer than 24 hours, we suggest adding a booster shot, which will help you to last another 24 hours. So we would repetitively add more booster shots of our labeling kits to help keep the clear image of the blood vessels during images. But typically it would be 24 hours.
Okay, he also has a following question. For the labeling, is it possible to tag other molecules with a fluorophore using IVIM's protocol, not just the antibodies?
Yes, we can definitely conjugate other molecules with our protocol. We've currently done some with proteins. And if you would maybe like to inquire to see if your sample is able to be conjugated with fluorophores using our protocol, again, you can contact us through email or you can drop us a Q&A on our website.
Thank you, Jenny. There was another question. We already gave the answer through email, but if you want to answer it again, once the chamber is implanted on the mice, how long it can be kept there?
So it would vary according to which organ you have implanted the chamber with. But if I talk about the cranial imaging window that I presented in my presentation, if inflammation is dealt with very well, the mouse can live up to at least 3 months and even up to a year with the window implanted in its brain.
Okay. If there are no more questions, we will conclude the Q&A session for today. And if you have any questions, you can also ask them through information@ibmtech.com. And thank you all for your active participation. As mentioned earlier, we recently launched the IBM shop where you can find and explore a range of imaging accessories, fluorescent dyes, and training programs for IBM imaging. And by creating an account, you will gain access to exclusive benefits that apply to your purchases. If you have any questions or need further assistance, feel free to reach out us at information@ibmtech.com or sales@ibmtech.com. And thank you once again for joining us. We look forward to seeing you at our next webinar. Thank you.